So hi, hello and welcome again, Microbehunter here. Look at this horrible looking orange, it's quite moldy, and today I'm going to show you how I've actually managed to grow the mold in a Petri dish so that I have a nice sample to put under the microscope. So why would anyone do that? Well, first of all, I wanted to observe how the spores of the mold actually start to germinate and start to grow, and I wanted to observe this under the microscope, and in the meantime I discovered something quite interesting, something that was a little bit unexpected, but I think I'm going to start at the very beginning here. A few days ago I found this orange in my refrigerator, and it was covered with a fungus called the penicillium fungus. It's actually a very common fungus that infects citrus fruits, and instead of simply throwing the orange out, I decided to try to cultivate the fungus that's growing on the orange. And in order to do that, I cut out some filter paper and I put some orange juice on the filter paper in a Petri dish, and I used a toothpick to carefully transfer some of the spores and some of the fungus from the orange onto the filter paper. And after a couple of days, it was really completely overgrown. It looked very greenish already, a sign that a lot of spores have already formed. And then I basically was quite happy because now I had plenty of samples to observe and put under the microscope. So what I did as a next step is I took a small sample here, mixed it with a little bit of orange juice, and then put it under the microscope with the hope of actually seeing that the spores will actually start to germinate and to start to grow the fungal cells. These cells are called the hyphae, and they basically should grow out of the spore, because that's the way that the fungus spreads over the air. The spores are scattered, and then when the spores land on a favorable medium, like for example other fruits, then they will start to grow, and then you basically are completing the infection cycle. And essentially I waited a couple of hours and not a lot happened really, so I was a little bit disappointed, also a little bit surprised, because why do the spores not germinate? But then after a day or so, I actually saw that on the borders of the cover glass, it started to become quite fuzzy. This was actually a sign that the fungus started to grow on the borders of the cover glass, which was kind of very unusual, I thought. So I put it, of course, under the microscope, and I could see that indeed the spores at the edges of the cover glass started to grow, but the ones in the center of the slide, they did not. And the answer is actually quite easy, and that has to do something with the oxygen supply, because the spores in the center of the slide did not get enough oxygen and could not germinate this way. And the spores that were found on the edges, on the border, of course, they could grow because they were in contact with the air. Of course, I also tried to do a little bit of a time-lapse video because I wanted to see whether the fungal cells actually do grow or not. And indeed, over the course of approximately 45 minutes to an hour, I was able to see that indeed the cells started to become longer and started to grow. I also realized that when you zoom into those fungal cells, that there is also quite a bit of movement going on inside those cells. Tiny dots are moving back and forth, and these are most likely vesicles and other cell organelles that are moving around inside the fungus. Yeah, so what do we learn from this here? Well, take care that the spores do not scatter around in your refrigerator or in the place where you store your fruits, because then of course they're able to infect other fruits as well. And if one of the fruits actually has become bad, make sure that you wipe everything properly. But before simply throwing it out, I think it's indeed a good idea to put it under the microscope and see a little bit on what's going on in our environment, because there are always new things to learn. I think I'm going to leave it at that. Hope that you liked the video. Of course, I would like to invite you to subscribe to this channel. Happy microbe hunting as always. See you around in the next video. Bye bye.